So my name is Megan Collins. I'm the research coordinator for GRIN, and today we are joined by three fantastic researchers who are going to talk to us a little bit about functional testing and what that looks like. Our first presentation is going to be by Dr. Ian Coombs. So if you want to go ahead and share your screen.
Yeah, thanks very much, Megan. Okay, let's see if we can do this smoothly. Okay, is that coming through okay? Yeah, great. Okay, so as Megan said, I'm Ian Coombs. I'm an electrophysiologist working at University College London, and I'm going to talk to you about how we can use expression systems to examine the properties of GRI variants to understand a bit more about them. I'm going to start just talking a little bit generally about the technique, and also some data that we've managed to produce looking at a novel GRIA2 variant. And so these expression systems, they're a really fast, efficient way of understanding really key biophysical details about the receptors, but it's actually the second step in the process. And the first step in the process is made an awful lot easier by the fact that we now have structural details of all of the glutamate receptors. So starting off with Ampa receptors back in 2009, then there were some NMDA receptors, kainate receptors, and finally last year we have the first structure of a GRID receptor as well. And so when we find out the details of a variant, it's very easy for us to kind of plug that into these structures and work out exactly where they are and exactly what they're likely to be doing. And the reason we can also work out what they're likely to be doing is that not only do we understand the structure very well, we also understand the mechanisms of how the receptors work. So they sit here in the postsynaptic membrane, ready to catch glutamate released by the presynaptic cell. And when the glutamate's released, it binds within this central region within the Ampa receptor, the ligand binding domain. And when this clamshell-like structure of the ligand binding domain closes, it pulls open the channel pore. And it's this opening that produces the electrical signals in the brain. And it's also these electrical signals that we can examine using our expression systems. So all of us researchers have all got all the glutamate receptor genes in the fridge, and they're stored within handy little units of DNA called plasmids. And these are circular little bits of DNA, and they make it very easy for us to amplify the DNA, purify it out, and then ultimately our goal is to use a process called transfection, which gets this plasmid DNA into our cell of choice, and then the cell's own machinery will turn this DNA into the glutamate receptors, which we can then examine. Another advantage of these plasmids is it's very, very easy to manipulate them. So anytime we find out the details of a variant, we can make that specific change in the DNA we already have. And it really does only take potentially less than 2 weeks from finding out the name of the variant to discovering exactly what this does to receptor function. So expression systems are incredibly useful. And once we've got these receptors on the surface of our cell, there's a couple of different ways we can examine them. So a more traditional way of doing it would be we put a glass electrode into the cell to record the electrical activity in that cell. And then we can just very slowly apply solution to the bath. And in this case, this is in the case of an agonist like glutamate. And as the concentration increases, the current increases, and we can understand, start to begin to understand details about how glutamate is activating the receptor. And we can also use these same systems to look at the effect of drugs, whether they be potentiators or inhibitors. And again, this can all be done very, very quickly once we find out the details of the variant. Now, the disadvantage of using this kind of approach is that this is not the way that the receptors in the brain see glutamate. They see it very, very short transient events when at the synapse glutamate is released. And glutamate is there and then gone in just a millisecond or just a few milliseconds. And so that is not the kind of thing you can do with this. This application takes seconds and minutes. And so to get a better idea of exactly how the receptors are going to behave in the brain, we go to the same situation as this, where we have access to the cell, but then we pull the electrode back. And what that allows us to do is get a tiny little bit of membrane on the end of our electrode. And because it's so small, we have very good control over the solutions that it can experience. And we use this device here, which has got 2 channels, so it's just a little glass tube. And on one side, we have a control solution, on the other side, a glutamate solution, and that allows us to really examine exactly how the receptors might behave in the synapse, going from a situation where there's no glutamate at all to a very high concentration of glutamate and then back. And it's this technique is the main technique that we use in the lab, and this is how we choose to examine the variants. So when we first were contacted by GRIN, we were kind of looking for a job almost. In the first question and answer session we attended, there was a question from a clinician about a particular variant in GLUA2, A643V, and they were wondering whether it was loss of function or gain of function, and this was going to dictate the approach they took with the patient. And so we decided to have a look at it, and it was immediately obvious by going through looking at the structural data that this was very likely to be disease-causing. So it was found in this completely conserved region of the glutamate receptors, this 9 amino acid stretch, and it was this final alanine here that was changed to a valine. And we know this is important because it's conserved, and this region is it's like the very it forms the channel gate. So basically this bit of the protein is all bunched together, the 4 copies, when the channel is closed, and when the channel opens they all spring apart, and that's what opens the ion channel that allows us to record electrical currents. And another reason we were could be convinced that it was likely to be disease causing was that for the AMPA receptors there's actually been another few variants been identified at this exact site. So for GRIA1, a threonine was found at this site, and for GRIA4 also a valine. And so the threonine variant has been looked at length and is known to be gain of function. And while it's likely that the valine was also gain of function, because they're similar amino acids, they're the same size and shape, even though their chemistry is subtly different. But there were 2 main options. Either this valine could destabilize the closed receptor, causing gain of function, or it could stabilize it, leading to loss of function. And we made the variant in our plasmid, we expressed it into the expression system and pulled a patch, and it was immediately obvious that this variant was gain of function. So by using a very brief pulse of glutamate, we can activate the receptor and then glutamate unbinds. And for the wild type receptor, this all happens within 1 millisecond. But for the variant, this was taking more like 10 milliseconds. And that was equally the case in the presence of the important auxiliary subunits called the TARPs, where the deactivation was slowed to 5 milliseconds, but then it was an additive effect with the variant, giving a deactivation time constant of 60 milliseconds. And this is enormously not how an AMPA receptor should behave. It certainly is likely to indicate an increased affinity. It binds glutamate longer, so it will be able to bind lower concentrations of glutamate and give a larger response. And this is likely to completely interfere with normal synaptic transmission. Okay, so we've kind of taken the structural information, we've made the gene, we've used the expression system to find that it's gain of function. And so the next logical step that you can also do with these expression systems is to try and find a drug likely to counteract the effects of the variant. So for AMPA receptors, the only prescribable drug, really, the only one that's widely in the clinic, is a negative allosteric modulator called prompinol, which has the potential to deactivate or to counteract the gain of function changes. And so to examine this, we looked at a slightly different application of glutamate. So AMPA receptors and most ligand-gated ion channels have a defense mechanism built in case in case the signal lasts too long, and they can turn themselves off in the continued presence of glutamate. For the wild type receptor, this desensitization was over 99%. However, for the variant receptor, it was only about 50%. So again, not only is there a deficit in closure, deactivation was greatly delayed, desensitization is also greatly affected. And this was similarly amplified with the TARP. And what we found when we applied prompinol was that prompinol could inhibit these currents. However, the inhibition was somewhat modulated and somewhat less. So for the wild type receptor, a concentration of 3 micromolar prompinol inhibited currents by between 80 and 90%. However, for the variant, this inhibition was down around 40%. And so what this tells us is that while prompinol is effective at inhibiting the currents, this inhibition is partial. And so and certainly you wouldn't design a drug like this. You would rather it have a preferential effect on the variant, as we heard earlier from Dr. Zhang talking about memantine and one of the GRIN variants. But this is suboptimal. So we intend to look further and try and find other potential AMPA receptor modulators that may have a greater effect on this variant. Just to further elaborate why it's important to do these kind of tests, we also looked at the threonine variant, which is also known to be gain of function. But the effect of the threonine variant was even greater than valine. It seemed to be, it was a much greater gain of function. There was no desensitization at all even in the absence of a TARP, and in the presence of the TARP, there was a very large leak current. So this channel remains open the whole time. And in actual fact, even in the presence of MBQX that should shut all the channels, still 10% of the current remained when the TARP was there. So while the threonine and the valine variants look very similar on paper, the actual manifestation of the change on the channel was appreciably different, even though both gain of function. Okay, and then finally, just a bit of mechanism. Going back to our structure again, there's a structure of the receptor with perampanel bound, and the site of the variant was immediately adjacent to this perampanel, and so any change here is likely to disrupt the perampanel binding site. And so it's very likely that this is the reason why perampanel was less effective against the variant. And further, the binding site only looks like this when the receptor is actually closed. As soon as the receptor opens, all of these bits around here move and perampanel will no longer bind. And that probably explains why, in the presence of the TARP at least, while there was a decent level of inhibition instantaneously, that inhibition decreased as time passed. Okay, so just to summarize, we used expression systems to establish the nature of an AMPA receptor variant, A643V, and found that while this gain of function was accentuated by the TARPs, and while perampanel can inhibit the variant, it seems to be less effective than against the healthy receptor. And what we intend to look at is other potential AMPA receptor inhibitors, and also to examine the influence of perampanel on other gain of function variants to see whether this is a universal property of perampanel on gain of function. Okay, thank you, and thanks very much to the Medical Research Council and to the team at UCL and also the clinicians at University of Michigan.
Perfect. Thank you so much, Dr. Coombs. So now we are going to pass the presentation over to Dr. Wollmuth, if you want to go ahead and share your screen.
So I'm going to talk again, sort of following up on Ian's conversation, but talking about functional evaluation of NMDA, disease-associated NMDA receptor variants. So again, as Ian sort of laid out, you know, an individual is identified with a missense mutation in a GRIN gene. Again, I'm focusing on NMDA receptors. We express this variant in heterologous expression systems, and we test function. And again, what's our goal? Our goal is to sort of identify whether it's a loss of function, whether this variant is causing a loss of function, that is, the protein doesn't work as well as it did before, or it's a gain of function. That is, it works too well. It works too well. And correspondingly, then, we want to bring in pharmacology, either positive allosteric modulators to enhance the function of the loss of function variant, or negative allosteric modulators to reduce the activity of these gain of function. And again, our goal is hopefully to normalize some of the function of the NMDA receptor, which in turn could normalize some of the behavior and activity of the individual affected by this. Now, the challenge again, and there's many of them, but one of them is that NMDA receptors have really a complex array of properties. So these are the properties that control their function at synapses. And so it's often difficult to define an NMDA receptor as loss or gain of function. And oftentimes these variants will produce in one parameter a gain of function and another a loss of function. So it's oftentimes difficult to define the appropriate pharmacology. And even if you define a variant as loss of function or gain of function, it's sometimes challenging to what to normalize. Again, what we want to do is to maximize normalizing this function. So one of the things we've been trying to work on is to develop an approach that can characterize these variants that requires a limited number of measurements, but also captures aspects of the physiology. Right? We want to capture this approach to capture the physiology of NMDA receptors. And then from this approach, these measurements we're going to make, we want to generate some consolidated parameters. So again, these consolidated parameters are going to contain a lot of information about these different biophysical properties, but they're going to contain it into one or two values, which will aid in allowing us to define them as loss or gain of function. And then finally, we want to use these consolidated parameters to help us guide the pharmacology. What sort of pharmacology can we use to normalize these, to maximize normalizing function? So again, we're using, it's a 3-step process. There's lots of steps in between that I'm not going to go into. So we're going to use pulses of glutamate to assay ion channel gating, and I'll discuss this in a moment. And then we're going to assay voltage dependence of magnesium blocks. So again, these 2 steps here encompass a lot of properties of the NMDA receptor. And we're going to do this 3-step process here. And from these combination factors, we're going to derive a charge transfer. The step 3, we're going to assay calcium influx using, again, and then from that, we're going to be able to drive a calcium transfer. Now, for NMDA receptors at synapses, there's 2 prominent roles they carry. One is a charge transfer, and this is their role in regulating membrane excitability. And another key physiological role of NMDA receptors is this calcium transfer. So again, we're going to derive 2 terms that describe charge transfer and calcium transfer. And again, then our goal is to use pharmacology, the appropriate pharmacology, to normalize this. And again, all experiments I'm going to show are with HEC293 cells. We're doing experiments with human GLUN1 and human GLUN2A. And we've selected a subset of variants, GRIN1 and GRIN2A variants, that are associated with epilepsy, some level of epilepsy. So I guess I got to stop, and I'm going to restart it. So for step 1, what we're doing is we're using actually pulses of glutamate. So again, what we're doing here is we're recording in the whole cell mode, and we're applying synaptic-like pulses here, very brief applications of glutamate to in this case, it's GLUN1 and GLUN2A. And we're looking at these pulses, these currents, and you can see there's various current amplitudes, they decay. But what we're interested in deriving here is the gray, this current integral, which is the charge. And so why are we doing it this way? Well, first of all, these pulses have physiological relevance, right? So at a synapse, NMDA receptors, as well as AMPAs, rarely see just a single pulse of glutamate, but rather they're going to see some pattern of presynaptic activity that reflects the activity of the presynaptic neuron as well as the release probability. So again, this charge or this current integral here captures a physiological component of dynamic NMDA receptors. I'm not going to go in detail, but it also turns out that this charge also captures a lot of all the biophysical properties of NMDA receptors. So a lot of the physiological functions of NMDA receptors are captured by this single term. Again, sorry, I got it. So here's just, this is wild type. It's just showing the response of wild type to these pulses. And here's just 2 example variants that I'm showing you. One is D552E. And you can see that in response to similar pulses like this, that the area under the curve is reduced. This variant, in terms of this parameter, is a loss of function. It cannot carry out this function as well as NMDA receptors, whereas this variant, N614S, actually shows a gain of function in terms of this parameter. But again, what's important to realize is that within here, this contains, that integral term there contains a lot of information, contains the gating properties as well as relative membrane expression. So again, but that again is only part of, I'm going to skip a slide just because I'm jumping in and out here, but that's only part of how the NMDA receptor is regulated at synapses. So, another part is the voltage dependence of magnesium block. So again, here's just wild type. This is a hallmark of NMDA receptors, that they're modified, that they're charged, that what they carry through the membrane at synapse is regulated by magnesium, and you can see that a variant affects this. Again, this is a single property, and here's all these variants that we've looked at, a large number of these variants. We see that some of them are gain of function and some of them are loss of function. Again, you don't have to worry about the absolute numbers because we're using this little color-coded chart here to indicate loss and gain of function. But again, this just reflects one of the parameters, right? Magnesium block. And what's important is not just magnesium block, but all those other gating parameters that I talked about before when we measured that integral. So then what we're able to do is take those gating parameters, we're able to put it into a modeled current now. We take the magnesium block parameters that we showed, so we're able to consolidate those all down into this sort of area under the curve here. And that area under the curve is what we refer to as the charge transfer. Again, it encompasses variations in current amplitudes, it encompasses receptor gating, and it encompasses magnesium block. So again, we have this one term, charge transfer, that encompasses all these factors. And again, here's just two examples of two variants, M641I, which is a gain of function, and N641S, which is an extremely large gain of function. And so then the third step is we assay calcium influx under using fractional calcium currents. It doesn't really matter. Again, we can derive then for the individual variants at a particular membrane potential, their effect on this ability to carry calcium. And but again, what we want to do is to integrate this calcium influx over a wide voltage range and with magnesium block and with all of the gating parameters. And so that's what's illustrated here. So again, this is the pulses in light gray. Here you can see that's the charge transfer. So that would be the sort of the impact of the NMDA receptor on membrane excitability. And then this darker gray is a calcium transfer. And again, those two terms. But now again, this is only at 1 voltage. And so we're able to, again, using model currents, to be able to derive the current under here as a term reflecting the charge transfer, and here reflecting the calcium transfer. So again, for each of the wild type and each of the variants, we can get these individual parameters to look at them. And again, here's just a summary of that. Again, you don't have to worry about the detailed numbers because you can just look at the loss and gain of function color scheme here. But we can see here's wild type for the charge transfer, here's wild type for the calcium transfer, and you can see we have loss and gain of functions. In some instances, we have no effect. And again, but these terms are encompassing a lot of the different properties of NMDAs. So now we just have these two single terms. And as a last slide, again, now the global goal is to be able to take the pharmacology, either these negative allosteric modulators or the positive allosteric modulators, to modulate NMDA receptor function to return it back to sort of a normal level. So again, this is showing the pulses for wild type. And you can see when we put on this NAM, this negative allosteric modulator called dextrofan, you can see it inhibits the function. And it does the same thing for these two variants here. But recall, these two variants here are gain of function. So their activity is greater than that of wild type. So again, so in the scheme down here, we want to normalize. So this is wild type. So this is what we're trying to get everything to, either for the charge transfer or for the calcium transfer. Again, these are gain of functions in terms of that. And you can see for at least at this concentration, we can get N614 back down to sort of that normalized level there, okay? And I'm going to stop sharing This was a very painful way to give a presentation, and I apologize for somehow not having my PowerPoint working right. Chaos worked, so it did work, and I apologize, but I'm going to stop sharing and let Graham take over now.
Okay, thank you. Can you see my presentation? Okay. Well, thank you very much. I'd like to thank Megan, Denise, and Keith for inviting me to talk. I was asked to talk about mouse models of GRIN disorders. I'm going to focus on the one that we have been studying as an example, but the principles will apply, although the details would differ for all of the different NMDA, AMPA, and kainate receptor variants. So our story started when I attended a GRIN meeting in Toronto 3 years ago. And I met Bryson and his parents, and Keith asked, is there anything we can do to help understand what could be the origin of the variant that is affecting his son Bryson? It was already identified as a point mutation, a de novo point mutation in the GRIN1 gene, a G620R substitution. And I had been in Toronto for a few years and realized that we actually had a fantastic mouse facility, the National Center for Phenogenomics, which is very good at making mouse models of human disorders. So I said that I thought there was a good chance that the TCP could make a mouse model. And indeed, fortunately, they were able to come through, and they made a single point mutation in the GRIN1 gene, substituting G for R, glycine for arginine. So I'm going to tell you a little bit about what we have done with this mouse, but of course the same could be done for any mouse model. It's just the outcomes would be, of course, different depending on the impact of the variants. So the first thing we found is that the mice were born. We never, and to this date, we have never seen a homozygous mouse, which means that it's almost certainly embryonic lethal if you carry 2 copies of the variant. But the heterozygous mice are born normally. This, of course, is the equivalent of the human condition. But they are lighter, they have a lower body mass, as you can see. And actually, this is a wild type, and this is the litter mate carrying 1 copy of the variation. And this is they grow, they seem to be totally viable, but they are definitely of smaller body weight. The sorts of things we can do is we can remove parts of the brain and study their physiology. In principle, we could do this to any part of the brain. In reality, we study the hippocampus. We study the hippocampus because it's particularly important for learning and memory, which is one of the cognitive domains that are affected in many of these GRIN variations. And also the hippocampus is an area we know more about than any other part of the brain. So it serves as a good model for the brain. We can look at the anatomy, and we have seen no gross anatomical differences in our mice. They appear identical to wild type mice at the level that we have analyzed. And then we can make these electrophysiological recordings. We typically take isolated slices of hippocampus, the so-called hippocampal slice preparation. We can position the electrodes to stimulate afferent inputs, and we can record from individual or populations of cells, such as here we're stimulating so-called CA3 pyramidal cells, recording from the CA1 pyramidal cells. So as we've heard in the previous talks, the NMDA receptors are very important. They're at synapses, and they mediate calcium entry and also charge transfer into the cell. But there are also AMPA receptors present at the synapse, and most synapses in the brain, including the synapse we work on, essentially operates by fast transmission working via the AMPA receptors, and under certain conditions, the NMDA receptors become engaged, where they trigger changes in the efficacy of the synapses. That is, they cause learning and memory to happen. So we have studied both AMPA transmission and NMDA receptor mediated transmission at these synapses. The first take-home message is that AMPA receptor mediated synaptic transmission is identical as far as we can see. We do not see any differences. And that is really good news, because that means that the synapses have developed normally. The connectivity, as far as we can tell, is absolutely normal. Even though they have had an impaired NMDA receptor function, this has not impacted on the way that the synapses have developed. However, the NMDA receptor mediated synaptic transmission is reduced, dramatically reduced. In fact, it's about 50% of its normal size. There are special tricks that I don't really have time to go into to explain how we can isolate these NMDA receptor-mediated synaptic responses, because normally they are blocked by magnesium, as we heard in the last talk. But if we remove the magnesium or lower the magnesium, then we can record these responses. So this is exactly what one might expect if you have a problem with the NMDA receptor and it's a loss of function. If it was a gain of function, then these responses would be bigger. So this is pointing to a functional loss of function of this variation. So in terms of what NMDA receptors do, they're normally being blocked by magnesium. They're not being active most of the time, but during periods of intense activity, the sort of activity that is now going on through everybody's brains as they're listening to me speak, I hope, is that you get transient activity, you remove the magnesium block, the NMDA receptors become transiently activated, and they cause persistent changes in how well those synapses operate. That is to say, the synapses are learning from their past experience. Because there is a reduction in function of the NMDA receptors, we would predict that this learning process is reduced. And the way that we and others study that is through a process known as long-term potentiation. And what we do is we study AMPA receptor-mediated transmission, which is pretty constant with time, deliver a brief period of high-frequency stimulation to mimic the learning episode, and this leads to this long-lasting increase in the size of the synaptic response. This is the so-called long-term potentiation, or LTP. In the mice, this LTP is still present, so these synapses will still have the capacity to learn, but as you can see, it's dramatically reduced. And again, it's about a 50% reduction in the synaptic efficacy. We can then go one step further and try and understand the underlying mechanisms of this effect. So this is the take-home message: LTP is reduced in the mice. In terms of the underlying mechanism, a reduction in function could be because there are less of the normal receptors at synapse, or it could be that there's the same number of receptors, but if they have a G620R variant, they are working less effectively, both of which would lead to a reduction in function. One way we've addressed this is to exploit one of the properties that had been identified previously using the hex cell type of recording that the previous speakers were talking about. This is from the Steve Trinellis lab, where they looked at the same variant and showed that in the hex cell expression system, you lose sensitivity to magnesium ions, which means if the receptors are getting to the synapse, they should have altered magnesium sensitivity. But if they're not getting to the synapse, then the magnesium sensitivity should be the same as in the wild type. And indeed, it's the latter case that we see. We've looked at the size of the synaptic response. This is the NMDA receptor-mediated synaptic response. We slowly increased the magnesium concentration from 30 micromolar to 1 millimolar, and we've compared the potency of magnesium to suppress the NMDA receptor responses in wild type, which is in black, and in the variant, which is in blue. And as you can see, these are identical, which tells us that it's not this situation, it's this situation. We're getting fewer receptors to the synapse, but the ones that we're getting there are behaving normally, as if the mutant receptors are probably getting broken down and not getting incorporated in the synapse. So that's good, because that means if we can boost the function of these receptors, then we can start to think about getting restoration of function. So we've in fact tried to do this using a number of drugs. The one I'm going to show you is memantine. We use memantine because it's clinically approved NMDA receptor channel blocker. You would expect it to be more effective in our gain of function, but when we did these experiments, it wasn't certain that it was a gain of function, because of course the lack of magnesium sensitivity could actually point to a gain of function. Bryson had actually been treated with memantine. Under normal conditions, memantine at a therapeutic concentration has very little effect on LTP. The reduced LTP unfortunately wasn't boosted with memantine. In fact, if anything, the reduced LTP was further reduced by memantine, suggesting that memantine would not be effective in this case. And indeed, what we would expect is that the most effective treatment here would be a positive allosteric modulator, and that is something we're now about to go on and investigate. I'm pretty much out of time, so I'm just going to show you one last approach, and that is, because these are living animals before we sacrifice them to take the slices, we can actually use some of the other mice to do behavioral experiments. And we've just started to do a behavioral phenotype of our mice. And one thing we've noticed is that the G620R heterozygous mice have less activity. And so this is tracking the activity of a mouse automatically as it's wandering around an open space. And as you can see, the variants are moving far less, and this is the quantification of the behavior. And they tend to walk more slowly, and they have impaired motor coordination. In addition, they have a number of repetitive behaviors which are autistic-like, to the extent you can get an autistic mouse. So you have an increase in repetitive behaviors, increase in grooming, you have an increase in sniffing. And there's probably many other differences. We haven't had the colony that long to look at everything, but we intend to look at learning and memory, anxiety, pain sensitivity, etc., etc. I'm aware that I'm out of time, so I've had to go through that very quickly. But the main principle is these sorts of methods can be applied to any of the variants. We just have to be able to make the mouse in the first place. So I just want to thank the people who have been involved in this work. The work I've described has mainly been done by Patrick Tidwell and Jinyo Lee, with contributions from all these other people. And I should highlight Laura Nutter, who is the person at TCP who was able to create the mouse for us. The funding mainly comes from Canadian government funding through the CIHR funding. Thank you very much.